Hello, everyone. Welcome to You've Got Potential, a podcast dedicated to undergraduate research and researchers and sharing knowledge about the undergraduate research community. Today, we will be talking with our wonderful co-host and student researcher, Monica, about her project on spinal cord injury. Monica and I met back in sophomore year, and she was the very first person I've ever met that was interested in pursuing degree in a graduate degree, more specifically a PhD. At the time, I was just like learning about it, and it was just so awesome to have someone to relate to. Over the years, we've really bonded over our love for research, and she's a wonderful person and wonderful student. So I'm so excited to hear about her amazing, super cool research today. And I'll leave the mic to her to talk a little bit about herself and for her to introduce her project to us a little bit.
Yeah, thanks, Georgia. She really gassed me up, but so yeah, I'm here to talk about spinal cord injury research that I'm doing. The 5-minute spiel on my research basically goes like this. So my postdoc found a synthetic peptide, which is just part of a protein that's made by a company. And that peptide improves functional recovery after spinal cord injury, only in female mice though. But even so, it's still a target for possible therapeutics. And so the problem with this peptide is that it can only be administered directly into the spinal cord because of different biological barriers, such as the blood-brain barrier that basically filters out any foreign particles that it doesn't want to access the brain. And so direct injection into a spinal cord is not really the most appealing treatment option for patients. And so my project is to find and evaluate compounds that can be administered intravenously or via the blood that would have the same effects as the peptide that my postdoc found. And so, so far, I found 5 compounds, and I call them mimetics because they mimic the structure of the peptide. And I'm currently evaluating if they also mimic the function of the peptide in cells. And eventually, we would like to transition into in vivo experiments and test them out in living mice. But coronavirus kind of messed up that schedule. And so instead, I'm working on the next step. So after you test something in vivo in mice, you have to determine if it actually made the mice better. And the problem with animal studies is that you can't just go up to them and ask, hey, are you good? Are you doing better, bro? No, we would all like to be Disney princesses, but we're not. So we can't talk to animals that way. So we have to be a little bit more creative in how we assess recovery. And so I started working in another lab at the Keck Center just last year. And we're working to develop a newer model to assess functional recovery, specifically in mice after spinal cord injury. And this new model is based off of machine learning, but I mostly work on the biology side. So I process and analyze tissue so that I can compare it to the scale that the computer algorithm is generating. And this is just to make sure that the scale accurately corresponds to the locomotion depicted in the neuropathy or in the spinal cord itself. And this can be hopefully applied later on in my original project, so I can evaluate if the treatment with the 5 mimetics actually improves functional recovery in mice or not.
That seems like a lot to throw on just an undergrad, especially when it comes to finding mimetics for a specific molecule. So how did that process go specifically?
Yeah, ELISAs basically. And we had an antibody that binds to the peptide, and therefore it's specific to that structure of the peptide. And so we then looked to see if other compounds would bind to the antibody. Remember in like high school biology when we learned like structure determines function? So we're going back to that. So we're making sure that we find compounds that look like the peptide, and hopefully they also mimic the function of the peptide. And so I feel like the reason why we do extensive functional tests after we found those 5 mimetics is because life is not like high school biology, right? And so nature kind of throws. Do you guys know the Is It Cake challenge? So they'll like make like a cake that looks like a cup, for example. And so the structure is like a cup. So you assume the function is like something that I can drink tea out of. But in reality, it's just something that you like eat and make people crazy on the internet about.
Yeah. You mentioned earlier on the talk, I want to get this right, that the polypeptide that you're researching had positive effects on spinal cord injury recovery only in the female mice, right? Okay.
Yep.
So I was wondering, like, why you think, like, just like a hypothesis on why that could be. Like, is the Y chromosome in males, does it transcribe for a protein maybe that methylates something on the X chromosome or something like that? I don't know.
Yeah. We're definitely looking into that mechanism right now. So like spinal cord injury is like male dominating condition. And so to find something that's like, oh, it works in females, it's kind of like a screw you to all you dudes out there. So like, so yeah, we're definitely trying to find out like how we can make this pathway work
Right.
for males. And so right now we haven't looked into a lot of the like methylation and genetic processes that you mentioned, but we did my postdoc looked into like androgen receptors. And so they didn't test it in spinal cord injured mice, they tested it in vitro. And one of the properties of this peptide is that it enhances neurite outgrowth. So a neurite is basically any process that comes off of a neuron. So these neurons are fairly young, so it's hard to tell like what's an axon and what's a dendrite. So we just all call them neurites.
Right.
And so the more it grows, that's just how we measure neurite outgrowth, which is important if you're like trying to rebuild a spinal cord, for example. And so they found out that if they inhibit the androgen receptor, then the peptide, like treatment with the peptide will bring back the neurite outgrowth in males, which it didn't before. So before, when they tested it just without inhibiting anything, then it only worked in females, the neurite outgrowth. And so the inhibition of the androgen receptor seems to bring back that property in males, but they were also looking at like the pathway. So cells basically communicate or like run the processes with something like a molecular tag. So it'll like tag protein, activate it, like, okay, you're it, now you do something, and then blah, 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 blah. And so they realized that they're not, like, if you inhibit the androgen receptor, it's not playing the same game. It's not activating the same proteins as the just the normal peptide treatment would. So somehow it's activating a different pathway. We're not sure how. We're hoping to figure out more with the grant. So we have more money
Well,
to
the
do so.
biology probably inhibiting such a major pathway like disrupts the cascade so much.
Yeah. So now that we've established what these synthetic peptides are generally supposed to be doing, what specific kinds of spinal cord injury are you studying?
Great. So the Keck Center really focuses on contusions, and contusions are basically like concussions for your spinal cord. And so they're like fast impact pressure. And so we have this machine, and I've never seen it actually done, but I understand the concept. So you would anesthetize a mouse, have it lying down, expose the spinal cord. So you'd have to do a surgery for this. And basically drop a weight down onto the spinal cord. That way we can control, like, if we want a moderate injury, a mild injury, or a severe injury, because those levels of severity are important in determining recovery, right? So there are other spinal cord injuries. So like there's compression, which is basically like if contusion is fast impact pressure, compression is just like slow. It's just like a slow squeeze. But we don't do that as much because what the population that we're trying to target is like mainly people who get into like car accidents. That's like a main driver or a main reason for spinal cord injury. And those are more often than not fast.
So you mentioned before that you're using a machine learning algorithm to try and figure out the effects of the molecules you're using on spinal cord injury recovery. Can you elaborate a little bit on that?
The way I like to describe the scale development and the AI as someone who's definitely not a computer person is that it's like Just Dance for mice. And like I used this analogy before because we actually use like an Xbox Connect as our camera. Yeah, as our camera to film the videos of these mice.
That's so cool.
Right? And then I was thinking about it more, and the analogy kind of goes further down into like how we process and train the program. So yeah, so we have a camera that is looking down into an open field, basically like like slightly larger than a Home Depot sized bucket that we like put mice into and let them scurry around in. And so the camera collects pixel data of like how close or far away the mouse is from the camera. So it's just depth. And from that pixel data, we can label like certain, I'm just going to call them dance moves because we're going to roll with this analogy. We can label certain dance moves. So the way Just Dance does it, or like the Connect does it for Just Dance, is that the data is labeled by people who like know what the dance moves are, right? Because the computer has no idea. So for example, like if the pixels show someone like, I don't know, clapping, I'm not going to dance because like I'm not going to embarrass myself or like put people through that. So just a clap, for example. Then it can identify that arrangement of pixels as a clap, and then it can tell the program or the algorithm, this is a clap. Like if you see a pixel, the pixels arranged in a certain way, that's a clap. And so now the model will recognize like, oh, okay, that's a clap. And so for mice, their dance moves are going to be things like rearing. So rearing is basically like when they stand up on their hind legs, like have their front paws up, and they're kind of like looking around to see like what the hell's going on. And so so far we've like labeled certain dance moves, and we've realized that some are going to be more predictive of spinal cord injury recovery than others. So for example, like if a mouse rears more or like stands up more, then it probably recovered better. because they are able to like support their weight on their back in order to stand up. And basically my side of the project is to verify that this model actually predicts recovery. So we're looking at the biological side and seeing what the spinal cord looks like. I think, 10 weeks after the contusion, so that surgery with the weight drop that I was talking about before. So a characteristic of contusions is that it kills like large myelinated axons, and myelin is basically like, if you're looking at like, I don't know, a cord, it's like the rubber part that's surrounding the wire to make sure it's insulated and the wire doesn't like short circuit or whatever. I'm not an electrical engineer, so don't at me. And so we're looking for demyelination for where the weight drops. So it's called a lesion site. So we identify the lesion site, and we're currently looking at the volume, like how big the lesion actually was, based on demyelination. And so we have antibodies that will detect like proteins specific to myelin, and that way we can identify like where the lesion is, how big it is, and so on.
So with your algorithm, how are you measuring the mouse? So for example, are you training the algorithm by just like giving it videos, or are you, before you give the video, you determine what like points on the mouse maybe will train the algorithm or something?
I'm not doing this, but I'm pretty sure that we train our algorithm based on like the height of the mouse at certain times. So because our camera measures depth, we can then translate that data into like a graph. So if, for example, if it's rearing and it like stands up, then the graph is going to be pretty like high, and then it'll come down quickly because it'll just like the mouse will basically come up and then come back, same as the graph. And then for like another behavior, if it like dives, for example, just like to go down, sniff something, and then come back up, then it'll go, down, up, and then back to normal. And so we'll see those, we'll also look at behavior, the videos, and like identify those behaviors, align it to those graphs, and then label those graphs as rearing, diving, whatever, tell the program this is what this is, and then like have it learn from there.
Yeah, that makes sense from what I understood, because then you're allowing the machine to collect what's objective data, and then you label that objective data as certain behaviors.
So I'm wondering then, what makes this more objective or reliable when it comes to replicating the work of human observers?
Great. So the current model that assesses a functional recovery after spinal cord injury in mice is basically like you gently place a mouse in like a kiddie pool sized bucket, and you have someone observe, like basically like with a clipboard and a piece of paper, like, oh, its tail is down, its tail is up, its head is up, it's walking like alternating paws, or it's like one at a time limping like that. And so like you have one or two people per lab that is in charge of this, and like they all get the same training, but there's human error, like maybe they're tired that day, they don't like see that the mouse like actually did alternate, or they missed something just because they were writing something down, or just in general, the variability between labs is going to be pretty high. And so you have like an opportunity to standardize this by creating a program that you can just feed videos into, right? Because that's how Just Dance does it. So like if the program learns from like a recording of me, like, oh, this is a clap, then it should also be able to recognize like if Georgia also claps, like, oh, okay, that's also a clap. And so it's just a way to like I said, standardize all of those features. And so like everyone across the country doing spinal cord injury can just like feed it in and hopefully have the same analysis.
Yeah, and it seems somewhat like your work acts sort of as a check on what the machine's learning, which
Yeah.
can be pretty valuable considering all the data it has to process and how long it would take a human to sift through all that. So, are there any specific regards in which you have to affect what the machine takes out of a given sample, or just check if its assessments are right with the tissue data?
So far I haven't been too involved with like the integration or the meeting of biology and the computer algorithm. So my work so far has been a lot of just processing. Yeah, so section, stain, and then image. So that process, like we say as if it's like 5 seconds and like, yeah, we did all this, but it takes so long. Like people underestimate like how much background or like work behind the scenes goes on in research. Like we just got a new undergrad intern in our lab, and like I'm helping training her, like, okay, this is what we do. And like this was the first week and she's like, yeah, I didn't realize how long this actually takes. So
And that's like the things that work.
yeah, no,
Like
exactly.
it's 90% of things that
besides
even work.
troubleshooting.
Actually, maybe it's even 99% that don't work. But
when you start out, it takes like 5 hours to do something that your postdoc does in like 20 minutes.
Exactly. No, for real. And like Georgia, the 99%, I invited my my second PI in the second lab that I joined to be a guest speaker at one of the events my club was holding. And she was talking about like how research is the only job that you can like fail 90% of the time or like 95% of the time and like still have a job with, which I guess is like it's cool to see like, oh, okay, like I can get paid to like not like have my experiments not work. But I feel like that might be an idealized version because they still do need to produce results to get funding and so on and so forth. So, but yeah, the room for error is larger than most occupations.
I would like to close or begin to close this interview with a couple of questions about specifically like your involvement with research at like your individual level. I was wondering how or like when actually did you realize that you liked research and that you liked it so much to want to pursue it as a profession?
I feel like it's not an aha moment. There's still times where I'm like, am I really ready for this? Like the imposter syndrome is definitely real. It's always been at the back of my mind since I started, because like going into Rutgers, I was like that pre-med kid. I went to all these like pre-med like clubs and like meetings, and everything was so overwhelming. And so I wanted to like find something else that I felt more comfortable in. And because I was in lab so often, like it felt like normal, and I was able to like build relationships, have like longtime friends, and like really dedicate myself to a project, which I think like is important to me, just to like be able to work on something and say that like, okay, I did that. And so yeah, I kind of deviated from my original plan of like going to med school, being a psychiatrist, and helping people with like mental illnesses. So now I want to tackle that large and rising issue of mental illness through the research aspect. So looking for different ways to like diagnose mental illness, or look for different predictive markers to like catch it early. And so that's hopefully where I'm headed after undergrad.
I wish you the best of luck with it, but I already know that you're gonna rock it. Absolutely. Like imposter syndrome is real, but you don't need it because you're a star. You really have so much potential, Monica. I would like to ask you, what would you say is the biggest pro of being both an undergrad researcher, but also like just a researcher? I mean, like you totally are both.
We talked about this earlier, but like the room for error. The room for error is also like room for growth, right? So yes, you can make mistakes, but you can also like learn how to grow from them. So like I remember, for example, just like lab processes, I took like those 2 hours or whatever that Venkat mentioned, like to make that dissection, and my mouse was like I don't know, like dried up and disgusting after perfusion or whatever. And like now it's like 15-20 minutes done. And now like I'm training the new undergrad, and she's just like, how are you doing this? I'm like, I don't know. Like you just kind of have to work at it. And it's just like a comfort level thing. And that happens with like all types of learning. So yeah, it's very easily translated into like other things that mean a lot to me. So like the learning curve, and like for example, I'm a figure skater. And so like learning something new, like I'm trash at it obviously the first time I do it. And then like I work at it and I work at it, and like it gets better. And so growth is pretty important to me.
I guess we can see you're a fan of the long-term potentiation.
Indeed I am.
Okay, thank you so much for your wonderful answer. I have one last thing to ask you, which is what advice would you give to a fellow aspiring neuroscientist?
I think the best way is to just like dip your feet into like big topics or like catchy topics. I think we mentioned this earlier, like in another episode, but like there's a reason why like things blow up on the internet, because they're interesting and they're catchy and they're applicable. And so like even just going on YouTube, finding like those, I think there's like a channel like 2-minute neuroscience or something like that, to just get like a baseline, or sometimes they go into pretty in-depth concepts, but they do it in a way that allows anybody to understand. And so just to get that background and be able to have a conversation with someone on like higher like levels of research later on is a good start to like one, understanding the content, and two, finding what you like about it, because not everything about neuroscience is going to be for you. Neuroscience is like one of those fields where it's so interdisciplinary. Like we have like we talk about ethics and stuff like that, because it's like human consciousness, and you have all of those like philosophical questions. You have like the biology side. And so because AI is based off of how our own brains work, they're super interconnected, and you can use one to study the other, and it's like a whole like meta loop. And so you can find your niche by just exploring like things on the internet.
Well, thank you so hugely much, Monica. It was very, very enlightening and very inspirational to hear about the wonderful work that you do, that you can learn so much about the human body, and how to cure it is just, I think, one of the most precious and noble aspects of research. And to see how passionate you are about it, it's really heartwarming. So I'd like to thank you again, and thank everyone else who was here and spent a lot of time to learn about your project and be involved in this discussion. We hope you stay determined, motivated, and inspired, because you've got potential.